Thank you for joining in today. Welcome to the quick demo on Mechanistic polylactide co-glycolide based long-acting injectable module. So coming to the introduction of long-acting injectable products. Long-acting injectables are in general intended for prolonged release applications. They are available in the market for the treatment of conditions like cancers, bacterial infections, and psychiatric disorders. Here the goal is to achieve less peak to trough concentration for a sufficiently long period of time. In a review paper by Reminar, you can find different tools and technologies used by the innovator companies to make LAA version of corresponding oral products of antipsychotic drugs. At formulation level, LAA products can be particulate systems. For example, polymeric particles, aqueous suspensions, they can be oil-based systems. They can also be solid or in situ gel forming implants. In polymeric systems, PLGA is the most commonly used polymer because it's a biodegradable polymer. You can tune the drug release by adjusting the lactide to glycolic acid ratio. Sometimes you can use a combination of polymers with different ratios and molecular weights. This is the PLGA model structure for in vitro release, where you have a solid implant in contact with in vitro release media. And a solid implant can be divided into two compartments, wetted and unwetted compartment, based on the degree of fluid penetration. In the case of PLGA implants, the polymer degradation and release are a result of a combination of multiple processes which are occurring simultaneously, such as fluid penetration, initial non-catalytic hydrolysis of PLGA, later autocatalytic degradation of PLGA. As a result of these processes, the long-chain polymer converts to oligomers. When the oligomer reaches a critical number, it would dissolve and diffuse out of the delivery system. As the polymer degradation continues, the porosity of the system increases and the trapped drug liberates from the unwetted compartment, and subsequently the dissolved drug would transfer from wetted compartment to the dissolution fluid. In the case of in vivo conditions, we would have additional processes like drug partitioning to local tissue and drug transfer from interstitial fluid to systemic circulation, which will depend on the drug physicochemical properties. In order to parameterize the model, the user need to enter the parameters related to compound formulation for running both in vitro and in vivo simulations. Anatomy and physiology is specific for in vivo. Additionally, for in vitro release, user can enter the release conditions such as solvent volume, pH of the media, sampling procedure, etc. The compound related parameters can be taken from literature or they can come from measured data. For example, solubility, molecular weight, log P. Formulation parameters such as lactide to glycolide ratio, solid fraction, size and shape of the implant can come from experimental data. Anatomy and physiology is already parameterized for both subcutaneous and intramuscular sites. User may use it as such or they can enter their own values if they have. In vitro testing module can be accessed from the ribbon on the top as shown it here. Once the IVRT module is activated by clicking on it within the substrate, you can browse FISChem and release subtabs to view and modify the parameter values and options. In FISChem subtab, you can specify the molecular weight, solubility, molecule type, etc. Whereas molecular radius is predicted from molecular weight. In the release subtab, the user can find the default values related to the compound, polymer and formulation, etc. And these are available for user to change. The user can specify the diffusion coefficient of the compound and release media, also the polymer related parameters such as rate constants related to hydrolysis and ionization. In formulation options, the user can enter the data related to formulation that is coming from characterization data. For example, lactide to co-glycolide ratio, porosity of the system, pore radius, polymer mean molecular weight and its distribution. Within implant parameters, user should enter the length, radius, and mass of the implant. This again comes from experimental data. The model parameters and outputs can be outputted to Excel sheets after the simulation is completed by checking them in vitro subtab of output options. Mainly drug concentration and mass profiles in addition to the polymer and implant parameters, for example, polymer mass, molecular weight, and porosity are available for selection. User can enter the experimental settings by accessing the in vitro design below outputs option. The options such as volume and viscosity of the solvent, surface tension, solvent pH, in addition to the sampling protocol, are available for changing. In this demo, I'll quickly show you how to access IVRT module. So this is SimSip graphical user interface. Here you can find IVRT icon. So when you click this icon, IVRT module is activated. Once it is activated, go to substrate. Here you can find two tabs, Physchem tab and Release tab. In Physchem tab, you can enter the physicochemical properties of drug molecule. In Release tab, you can modify the existing values for compound specific parameter, that is diffusion coefficient of compound in solvent, polymer related parameters, formulation options, and parameters. So here you can enter the mean molecular weight and the percentage CV on the mean molecular weight in a distribution. So here you can enter the implant parameters like length, radius, and mass of the implant. If you go to the in vitro design, here you can change the experimental settings. So the settings can be modified like dosage of the formulation, duration of experiment. So two different sampling protocols are available. Every 1 hour or every 2 hour continuous profiling. User also can define specific time points in order to get the outputs. So within outputs, you can find different sheets. You can output LA parameters, drug concentration and mass profiles. And apart from that, you can also output polymer degradation profile, polymer mean molecular weight profile, etc. So when you click on these PK profiles, they will be outputted into your Excel sheet. Additionally, in Excel options, you can overlay observed data file if you have an XML file with you. In this demo, I will show you how to access SimSip plugin for long-acting injectable module. So if you go to add-ins and click on the SimSip plugins, you will see different options. So you have to click on generate PE data entry template here. Select that one and select PKPD profiles. As you can see, once it is selected, PKPD profiles are selected, you will see a data entry template. So here all you have to do is change the dependent variable. So you can select drug released from implant in this drop down menu. Change the unit to percentage. Then enter the subject ID. Here the subject ID is your sampling ID. So you can mention the subject ID here. You can also mention the time. What is the time of collection of your sample? By default, the time is in hours. So here in DV column, you can enter the percentage drug release. So the DV ID is nothing but this one, DV ID. Here in this case, it is percentage drug release from implant. So here you can enter the DV ID. You can apply uniform weighing. If you have standard deviation or standard error, you can also enter that. So here you can you can select whether it is a substrate or inhibitor from this drop down menu. You can select substrate now. From route of administration, select long-acting injectable. Dosing unit, what is the dose? So now I'm selecting mg. The amount is 1 mg here. So now all I have to do is to go back to add-ins, go to SimZip plugin, save SimZip PE file. Once you've done that, if there are any errors, it will show you this error message. So you have to rectify that. So here my error is what is my subject ID. So once it is rectified, I can go back to SimZip plugins and save SimZip file. So the writing is completed. Once the writing is completed, you will see a spaghetti plot like this. Coming to the accessing the in vivo simulator for LA PLGA implant model. LA module in SimZip simulator can be accessed by selecting the long-acting injectable and dosing subtab of trial design. Once it is selected, a pop-up menu will appear asking you to select the physiological injection site from the drop-down menu. Currently, we have three different options. Those are subcutaneous, intramuscular, and other site. Similar to IVRT module, in the absorption screen, the user can access the compound parameters and formulation specific parameters. However, we have local tissue related parameters such as partition coefficients and diffusion coefficients in the in vivo simulator. At present, two different formulations can be modeled using the LA module. Those are solid implants and in situ gel forming implants. Either of the two can be selected by the user by checking on the screen. Additionally, you can run parameter estimation to estimate the value of a model parameter from the observed data. You can also use sensitivity analysis to assess the impact of parameters on pharmacokinetics. This is also applicable to in vitro module where your dependent variable is percentage drug released from the implant. Explore the link shown here to know about fitting, and if you have SimZip license, you can find videos about parameter estimation and sensitivity analysis in members area. In this demo, I will show you how to access the SimZip in vivo simulator for long-acting injectable module. So this is the graphical user interface. If you click trial design and go to dosing, here you have to change the route of administration to long-acting injectable. Once you select long-acting injectable, it will ask you for specifying the physiological injection site. You can select one of the site from this drop-down menu. For now, I'm selecting subcutaneous. And if you go to population and browse local injection site, here you can find the physiology. You can check different physiology like subcutaneous, intramuscular, or other site. If you have specific data related to this physiology options, you can modify them and use them for running the simulation. If you go to substrate and absorption screen, here you can find different parameters related to drug, polymer, and formulation. So these drug parameters are partition coefficients and diffusion coefficients related to the in vivo model. Here you can see the partition coefficient between interstitial fluid to lipid, and this one is split to interstitial fluid because these parameters are required for in vivo simulation, whereas not required for in vitro simulation. So within these polymer parameters, if you want to modify any of these parameters, you can click on the padlock. So it will open and you can modify the parameter value before running the simulation. Formulation options, here you can see two different options are available. By default, solid implant model is activated. If you want to run a simulation for in situ gel forming implant, you can simply check this one and run a simulation for in situ gel forming implant. Also for drug particles, if you want to activate dissolution model, you can do that by checking it here. Also, if you want to modify this mean molecular weight of the polymer, you can modify it here, and you can see the distribution as well, how the monomer distribution is there by pressing this view distribution. So implant parameters can be found here. You can modify or you can specify the specific values of yours. The implant parameters are like length of the implant, radius of the implant, mass of the implant. Once you parameterize the model, you can go to outputs, and within outputs in sheets, you will select different profiles, drug profiles, polymer related profiles, and implant related profiles to output. So once outputs are selected, you can run a simulation here. After simulation is completed, an Excel file will be generated. Within the Excel file, you can browse for the results. This is an Excel file generated after running a simulation for a long-acting injectable module. Here you can see in summary the route of administration, dose. If you go to sim healthy volunteers, you can find the physiology of local injection site. Here you can see the outputted values for different physiological parameters. If you go to concentration profile, CSIS, this is a systemic concentration profile of drug. So this is a mean profile, whereas this one is 5th percentile and 95th percentile of systemic concentration. So in LA parameters tab, you can see the outputs related to formulation. So these are formulation related outputs, and this is physiology related outputs. We also have outputs related to PhysChem. So here you can get the different parameters in this tab. So one thing it is noteworthy to mention is like we do not have variability currently implemented in formulation. Here you can see the formulation parameters are exactly same. They are not different for different individuals. So this is mass profile. Here you can see the mass profile in unwetted compartments and wetted compartments. So we also have a profile of mass that is dissolved in solid implant compartment. So this is the one. This is in interstitial fluid compartment, the mass profile. We also have mass profile in local cell compartment. Similarly, we have drug concentration profiles for different compartments in these outputs. Additionally, we also output percentage drug released as a function of time. Also, drug release rate. Here you can see on y-axis, this is the release rate versus time. We also output polymer mass. Here you can see the mass, how the mass is changing as a function of time. In terms of lactide and glycolide, we also have an additional output for total polymer mass as a function of time. So this is a percentage degradation profile for polymer. Here you can see the polymer degradation as a percentage with respect to time. We also have a polymer mean molecular weight profile as a function of time, and porosity, what is the porosity or solid fraction as a function of time. We also output water occupation within solid implant. Here you can see wetted radius as a function of time. And this graph is about percentage occupation. What is the percentage of water that is occupying the solid implant? We are additionally also output pH within the compartment, within the implant compartment, intra-implant compartment, and this is in the interstitial fluid compartment. Here you can see how the pH is dynamically changing as a function of time. In summary, the model considers measurable critical quality attributes to explain the drug release. In general, modeling of in vitro release can facilitate understanding the behavior of a delivery system. Additionally, one can perform and test the in vitro to in vivo extrapolation based on the availability of data. Lastly, once the PBPK model is developed, it can be used for a specific application. For example, running a virtual bioequivalence to compare test versus reference product for generic drug approval. I hope you are now familiar with the mechanistic polymer-based long-acting injectable module within SimSip Simulator. Thank you very much for listening.